Hello and welcome to an introduction to cell division, part 3 of 3 for the Alberta Science 30 curriculum. This covers topics in Unit 2 or the biology unit. So what are some differences between normal cells and cancer cells? Normal cells reproduce exactly as they're meant to, whereas cancer cells keep reproducing and they don't know when to stop. So we call this abnormal mitosis. In normal cells, they stop reproducing when they're supposed to, so they don't overgrow or make a tumor or a mass. If they are damaged, normal cells are destroyed or repaired. In cancer cells, they don't die if they move or metastasize, so moving is called metastasis, into another part of the body. Normal cells tend to stick together in the place that they're meant to be, so liver cells are found in the liver and lung cells are found in the lungs. They specialize or mature properly. Cancer cells don't tend to stick together. They like to move around, they break off, and they move to other areas of the body. And they don't specialize, they stay immature. So here's some pictures of skin cancer, and here are some ways you can sort of see if you have skin cancer. You can see an asymmetry. You can see irregularity in the border of the cell. The color is off, so it's dark. And they're large in diameter, so if they're 6 millimeters or larger in diameter, you should get them checked out. Here's an example of a cancer cell moving around or metastasizing. So you have a cancer that starts in one location, and what it tends to do is it tends to grow and to spread out. These go into your circulatory system, and they get moved around through your blood. Where they get lodged in other organisms, they get lodged in other organs. For instance, in this case, it gets lodged in the lungs, and when they're in the lungs, they start to grow and divide, and they cause more tumors. They move from the place where they started to other areas of the body. What are some commercial applications of mitosis? Cloning is an application of mitosis. This is the production of identical offspring from a single parent cell. A natural form of cloning are identical twins. Identical twins form from a single zygote cell, or a single fertilized egg somehow splits and produces 2 babies instead of 1. It's a form of asexual reproduction. Cloning was originally done commercially by taking plant cuttings. So taking bits of plants and trying to root them. It's advantageous because the parent provides nutrition, it's quick, and it doesn't require a partner. The disadvantage is that it has cloning results in a limited gene pool. Offspring are identical to parents, and limited genetic variation. If there's a mutation in the gene pool, those mutations are passed on. Plant tissue cultures and cloning laid the groundwork for genetic engineering. So here's an example of simple cloning. You have a parent carrot. They've taken a single cell and a bit of the carrot and have put the proper hormones or treated it with the proper hormones and put it in the dirt. And a new carrot is formed. So this new carrot is genetically identical to the old carrot. Asexual reproduction is the formation of new organisms from cells of a single parent. It's very common in plants. The offsprings are always genetically identical to the parent. Examples include strawberry plants sending out runners. Those runners are genetically identical. Amoebas divide into 2. So an amoeba is a simple eukaryotic organism that divides into 2 via a process called binary fission. An organism called a hydra forms new hydra by budding, so releasing a small organism out off of the parent organism. Some animals can reproduce by a process known as parthenogenesis. Parthenogenesis is the production of offspring that are genetically identical, that don't have to be reproduced by or produced by sexual reproduction. Mushrooms release spores. Asexual reproduction first sparked the idea about cloning experiments. So people started to think, if this occurs naturally, can we make it occur in a lab, and can we profit from it? So here's some strawberry runners as an example of asexual reproduction. Here is the bud of the hydra. And the most famous, when I was going through science classes, cloning experiment, was the production of this sheep called Dolly the sheep. And here's how this experiment happened. They extracted a mammary cell from the sheep, and they put it in nutrient deficient the media, or a sterile solution to keep the cell alive, but to stop it from undergoing a cell cycle. They took the nucleus, or in the meantime, they took an egg from another sheep and extracted the nucleus from that egg. So they took the DNA out. Then they fused this cell containing this source DNA from this other sheep, and they put it into the egg of the donor egg that they took the DNA out of. Then they gave this cell an electric shock, which opened the cell membrane and triggered cell division. It sort of tricked this egg cell into thinking that it had been fertilized, and an embryo was formed. This embryo was allowed to grow to this blastula stage, and then it was implanted into a surrogate mother. After a 5-month pregnancy, a new lamb was born that was genetically identical to this one. They called this lamb, or the sheep, Dolly. So they took out the 1N nucleus and they replaced it with a 2N nucleus. So the haploid nucleus was removed from the egg of the surrogate sheep. The diploid mammary cell nucleus from the animal to be cloned was inserted into the egg nucleus. An electric shock was used to trigger cell division. The embryo developed in vitro, which means in a test tube, and at the blastula stage it was implanted into a surrogate mother. The end result was a genetically identical sheep was eventually born. What's the difference between identical twins and fraternal twins? Identical twins are the result of a single fertilized egg dividing, and fraternal twins are the result of 2 eggs being fertilized, released into the uterus and being fertilized at the same time. Identical twins arise from a single egg fertilized by a single sperm. During mitosis, a single cell breaks free and a second embryo develops. The offspring have the same sex, blood type, and genetic makeup. In fraternal twins, these are the result of 2 different eggs being fertilized with 2 different sperm. They do not have the same genetic makeup, and they are no more similar than regular siblings. They share a uterus. They are not clones. So here you have identical twins who are the result of 1 fertilized egg splitting, and here you have fraternal twins.